Hello, everyone, and welcome back to our Chapter 5 lecture over eukaryotes. This lecture is going to cover what a eukaryotic cell is, some of the traits that are found in eukaryotes that make them eukaryotes and that are different than prokaryotes, and things like that, and then just some of the main categories of eukaryotic cells that are found out there. So let's go ahead and get started with that. So the very first eukaryotes on the planet appeared around 2 billion years ago. So life on Earth has been around for about 3.5 in the form of prokaryotes, a little longer than that probably. And then our very first eukaryote showed up about 2 billion years ago. So the origin of eukaryotes is very cool. The theory behind this is called the theory of endosymbiosis. It's pretty neat. You have very primitive prokaryotic cells that are swimming around in the ocean, and they come in different sizes, bigger ones, smaller ones, things like that. And that's just kind of how nature works. That's what we see today, bigger, larger size cells. And cells eat each other all the time. It's what they eat for food. So big cells eat smaller cells. That's just how it works. And sometimes those big cells don't necessarily digest the smaller cells that they eat. So when that happens, the smaller cell inside can now get a nice free ride around the world. And it doesn't have to worry about being eaten because it's already been eaten, and the big thing's probably not going to get eaten again. And it gets so the big thing also eats food for it and finds food, so the little guy doesn't have to go do anything. And then the big guy, he has this little guy living inside of him, but the little guy doesn't have to waste any energy, going to survive in the world. So all the extra energy that little guy makes. The big guy can then use, which is pretty cool. So all the extra energy that the little guy doesn't need. So this is how specialized organelles and things started to arise in early eukaryotes. So these were two prokaryotic cells that started living together and kind of went from there. Evolution took over. The origin of things like the endoplasmic reticulum and the Golgi apparatus and things like that are folding of membranes to increase surface area on the inside of the cell. So if the cell needed more space to do things, you can't just make a cell bigger because eventually you need more food. So the bigger the cell, the more food you need. Eventually you run to a point where you can't get enough food, so cells can only get so big, kind of thing. So instead of just getting bigger, they would just add on more layers on the inside of their cells, and that's what they do. So they just fold the cell membrane a lot. So that's how they added more space. So that's the endoplasmic reticulum and the Golgi apparatus and things like that. So endosymbiosis is the really cool theory that's been put forth to explain the origin of eukaryotes from prokaryotes. And it took, as you can see, quite a long time, about 1.7 billion years, for that to occur after prokaryotes once appeared on the planet. So these are some of the very first eukaryotic cells. You can see that they're quite large compared to bacterial fossils, for one thing, but they have specialized things inside of them that we recognize as chloroplast. Now, if you recall back to the size of the microbial world, mitochondria are about the same size as bacteria, and this is why. So chloroplasts are about the same size as bacteria, and this is why, because that's what they are. So ancient bacteria that moved inside of a larger bacteria eventually became mitochondria. Mitochondria have different size, have their own ribosomes, which is really cool. They're smaller, they're 70s, the same as a bacteria. They have a different set of DNA inside of them. They divide independently of our cells. They're the same size as bacteria. They are bacteria. And that's the endosymbiosis theory in a nutshell. The chloroplast is the same thing. An ancient photosynthetic bacteria was uptaken by another larger bacteria, and that became the chloroplast. So really cool origin stories or theories for the early eukaryotes on our planet. So these are some of the characteristics of the eukaryotes that we'll talk about in this lecture. Protozoans, I've talked about them a little bit. The weird guys that are kind of in between our eukaryotes and our prokaryotes. They're all eukaryotic though, but they have very primitive eukaryotic features. They're all unicellular organisms, but there may be some that are colonial dwelling. But that, if you recall, that means that they live together, but they don't need each other to survive, kind of thing. Fungi. Fungi are studied a lot in microbiology. Fungi can either be unicellular, multicellular, or colonial. And then algae, same. And then we've got our weird guys that are kind of tossed in microbiology, but they're macroscopic, the big ones. You can see an adult tapeworm. You can see adult arthropods, things like mosquitoes and things like that. But the larval forms of the parasitic worms, tapeworms, and things like that, you can't see without a microscope. So the eggs and things like that, the larvae. And that's why they're put into microbiology. So those guys are also eukaryotes. And our arthropods, things like mosquitoes that move around, organisms that carry disease, or that like malaria, Rocky Mountain spotted fever, ticks, and things like that. They're also going to be lumped in there as well because they are part of the microbiological transmission of particular diseases. So this is supposed to be a picture of our eukaryotic cell. You can check that one out on the PowerPoint. But we're going to work the same way that we did with the prokaryotic cell, from the outside in, with our eukaryotic cell. So on the outside of our eukaryotes, before we get to the cell membrane, you will find things that move the eukaryotic cell around. So if you recall, prokaryotes have a flagella, the little whip that moves them around in the environment. Eukaryotes have a flagella as well. But the flagella in a eukaryote is extremely different than the flagella in a prokaryote. In the same way that a jet engine is the same to a prop engine or propeller, they both allow planes to fly, but the way that they do so is drastically different. And this is the flagella to a eukaryote and a prokaryote. They both make the organism move, they both flap around in the same way, but they're not put together at all the same way. So the eukaryotic flagella is extremely complicated compared to the prokaryotic flagella. It's put together in something called a 9+2 arrangement, which you can see here. There's 9 doublets here with 2 single couplets in the middle, and these are microtubules, things that make up the cytoskeleton and things we'll talk about in a second, make up the flagella. So 9 sets of 2, and then 2 singles, or 1 single and then 2 of those, I guess, in the middle of the flagella. So 9+2. Whereas a bacterial flagella just has 1 large whip inside of it. So it's a lot bigger than the eukaryotic, or than the prokaryotic flagella as well. The eukaryotic flagella you can see sometimes underneath the microscope. They're quite large. And the other difference is it's covered in a layer of cell membrane. So you can see this here. So the eukaryotic flagella, the cell membrane goes all the way around it. So you can see that here. So everywhere that the flagella is going to be, the cell membrane from the rest of that cell is going to just kind of extend around it and cover it up as well. Whereas the prokaryotic flagella does not have that. Another thing that will be found on eukaryotes that help move them around or do things for them that are outside of their cell are going to be cilia. Cilia are found on lots of cells inside the human body. They are tiny little flagella-like structures. They're not flagella, but they kind of work the same way. They just kind of whip around. And then they just go in one direction like this. They have a power stroke where they move things, and then a recovery stroke. So you can swim forward, you push them down, and then you kind of relax them back. Swim forward, swim back kind of thing. And that's how organisms move. They just kind of swim along in the environment. In our body, our cells have them. They live. They're found on the cells that line your respiratory tract and your intestines and things. And they help move things that you swallow that are bad for you, or inhale, like mold spores and bacteria. It helps move them out of your respiratory tract, and it helps move food down the digestive tract. But in organisms like protists and things like that, it helps move them around in the environment. So they use their cilia, kind of like little tiny flagella, to help swim around in the environment, and they can also help them move food into their little mouth hole. They don't have a mouth; they have things called a gullet, a big open hole. And they just sweep the food in there. They're kind of like little brooms. They're only found in eukaryotes, which is kind of cool. So they're not found in prokaryotes at all, and they're kind of rare in the animal kingdom, other than just a very select group of protists and cells inside of us that I mentioned earlier. But they're pretty neat little things that are found only in eukaryotes. So next we're going to talk about the glycocalyx, and that's essentially the thing that's right underneath the flagella, right above the cell membrane, or above the cell wall. In the exact same way that it was found in prokaryotes, the eukaryotic glycocalyx is the exact same thing. That slime layer, that capsule, it's going to allow the cells to stick to things. It's going to allow them to have that little extra layer of protection from the immune system, and it's going to allow them to talk to one another. It's the exact same concept. as it is in our prokaryotes. Not every eukaryote is going to have a cell wall. We've talked about the types that do. Fungi have them. It's found in the form of chitin, which is a very thick polysaccharide. Cellulose is mixed in there as well for certain species of fungi. And then you have plants that have cell walls, which are made out of cellulose as well. Algae have the same thing. Some of them do, some of them don't. And what makes them up is a little different as well. But some eukaryotes have cell walls. Animal cells don't. But plants, fungi, and algae do. So the inside of our eukaryotic cell, once we get right underneath that cell wall, the cell membrane, is going to have the same thing that makes it up as our prokaryotic cell membrane, that phospholipid bilayer. It's made up of 2 layers of phospholipids, one on top, one on the bottom, with the tails in the middle, and a bunch of surface proteins and transport proteins and bridging proteins and things that help influence the movement of that cell membrane, keep it fluid. They also help to allow the movement of ions and big things that the cell might need in and out of its cell. So it's selectively permeable. This is the diffusion concept, that it allows certain things in and not other things, so the cell can control a little bit of what it wants inside of its cell, so it doesn't get too much or too little of a certain thing. And the eukaryotic cell is also going to have cell membranes around the organelles inside of it. So the mitochondria, the Golgi apparatus and things like that, they're going to have membranes that surround them. So you're not just going to find membranes surrounding just the cell. You're going to find them on the inside of the cell as well, surrounding organelles. So lots of different places there are the membranes. The nucleus is surrounded by one as well, called the nuclear membrane. So membranes are found throughout the cell, and they separate off different things and they keep things protected. So membranes do lots of different things for the cell. So the nucleus, you've probably heard this one all throughout your life, that the nucleus is the control center or the brain of the cell. That's pretty much what it is. It's where all the DNA is going to be protected. It's where the DNA is going to be found. It's all going to have the DNA in the middle, surrounded by something called the nuclear membrane, which is another cell membrane, phospholipid membrane. And it's going to have the protection quality to keep the DNA and things safe inside. Where all the DNA is going to be found and protected from the environment. Well, inside of that nucleus, you're going to find a little blob called the nucleolus, and that's going to be where RNA synthesis that makes ribosomes and where the ribosomes are going to be put together is going to occur. So ribosomes are going to be birthed there. They're going to be birthed inside the nucleolus. They will travel inside the nucleus to the outside of the nucleus through these little holes called the nuclear pores, through this nuclear membrane, and out into the cell cytoplasm. Once they travel into the cell cytoplasm, they will either stick to the rough endoplasmic reticulum, which we'll chat about in a second, or they will just freely float around in the cytoplasm of the cell, waiting for an mRNA to find them where they can make a protein. So the nucleus and the nucleolus. So the endoplasmic reticulum on the inside of the cell, you've heard this one before, comes in 2 forms: the smooth endoplasmic reticulum and the rough endoplasmic reticulum. The rough endoplasmic reticulum is going to be bound to the nuclear membrane. It's going to be sticking out of it. It's going to be found snaking around inside the nuclear membrane through the cytoplasm. And the ribosomes are going to come right out of the nuclear pore. They're going to stick to it. And it's going to get the name rough because it's covered in ribosomes, just waiting for mRNA to stick to them, where proteins are going to be made. And it gives it a rough kind of a textured look. So that's why it gives it like a cobblestone road, I guess. And that's why it gets the name rough endoplasmic reticulum. So mRNA comes out, it's ready to be processed, and then there's a ready factory for it right there on the rough endoplasmic reticulum, just waiting to go. So that's where proteins are going to be made for the most part. The smooth endoplasmic reticulum is going to process things inside of the cell, so nutrients, things like that. It's going to make certain lipids and things like that. It's going to store lipids. It's also going to send them around inside of the cell to the cell membrane and things like that. So the smooth endoplasmic reticulum does not have ribosomes bound to it, hence the name smooth versus rough that does. So you can see here the concept of making proteins. For our ribosomes, they come right out of the nucleus to the nuclear pore. Directly, the little ribosomes, they bond to the rough endoplasmic reticulum. The mRNA does the same thing, straight out. If the nucleolus are right onto a ribosome, instantaneously can be pumped right into the rough endoplasmic reticulum, which can then package it, ship it around, and do what it needs to do. So a really cool way to instantaneously kind of streamline the process. So the Golgi apparatus is going to be a very flat blob of kind of stacked membranes called cisternae that's going to modify proteins around, store them, and ship them around inside of the cell. So a protein will come in the top of the Golgi apparatus that needs to be changed to work better or to modify to do something different inside of the cell. And as it travels through the different little layers of the Golgi apparatus, the Golgi apparatus is going to modify it or change it around. And when it gets to the end of it, the Golgi apparatus will then send it where it needs to go. So the Golgi apparatus is pretty cool when it comes to shipping around proteins and things like that. Moving things around inside of the cell can happen lots of different ways. You can move things in and out of the cell with vesicles. You can move them with the Golgi apparatus using a vesicle to send them around. So lots of different cool ways that cells can move things around inside of them. Inside of cells, you will also find other little membrane-bound things that are not necessarily going to function to make energy or things like that. These are more meant for breaking down particles, storing things, or eating, stuff like that. So a lysosome. So lysosomes are little membrane-bound balls of digestive enzymes that are going to be found inside of eukaryotic cells. And when a eukaryotic cell comes in contact with something it wants to eat, it's going to engulf that food particle. And it needs a way to digest it. Well, they don't have teeth or a stomach like we do. So essentially they're going to throw a little water balloon, a lysosome, of digestive enzymes at whatever they've eaten. That digestive enzyme will burst out of the little water balloon, the lysosome, wash over the little food particle and break it down. Now, if they eat a bacteria or something like that, if they're infected, the lysosome can kill the organism, the bacteria that they've eaten. So it can also help to prevent infection. So a lot of different functions there like that. So lysosomes are pretty cool. Vacuoles can also be found inside of cells. Not every cell is going to have a vacuole. Some have more than one. They do different things. But they can be used for storing waste products or storing water, storing food, nutrients, and things like that. So it's essentially just a big giant membrane-bound sack, kind of like a canteen, that they can fill up with things that they either want to get rid of or things that they want to keep. And they might sometimes have one that does both. So it depends on the type of cell. And when you have a phagosome, which is a vacuole that's merged with a lysosome, so it's a digestive vacuole pretty much. So they have a bunch of food they want to break down, they bond a lysosome to it, and then they can break it down and get rid of it inside of there. So that's a pretty neat, cool way to do that. So they essentially have a bunch of food store, and they can break it down inside of the storeroom and not have to do anything else. So a pretty neat way for them to quickly get rid of their large food storage. So the mitochondria. We've talked about the mitochondria already. It's pretty much a tiny little prokaryotic bacteria that lives inside of our cells that provides energy for them. They have their own DNA. They have prokaryotic ribosomes. They make energy. Essentially, that's their job. The mitochondria is the powerhouse of the cell. You've heard that a million times. It's folded on the inside in the same reason that the endoplasmic reticulum and the Golgi apparatus is, because it needs a lot of space to make energy. So instead, you can't go bigger, but you can fold the inside. And that's what happens. So the cells just fold up the inside of their cell membranes to give extra space on the inside for the energy production to occur. And that's essentially what happened inside of this tiny little bacteria. And then over billions and billions and millions of years, it has eventually just evolved to coexist inside of our cells and function as an independent organelle instead of a bacteria that it once was. So they're pretty neat. They divide independently of our cells. They have their own DNA. They are essentially still a bacteria that just kind of lives inside of our cell. Chloroplast, exact same thing. It's the mitochondria, but for plants. It produces energy via photosynthesis. They're found in algae cells, plant cells, things that are green, that use photosynthesis via the sun. So inside of that, you'll find little stacks called grana. They essentially look like little stacks of quarters, little things called thylakoids that are stacked up into a big stack called a grana. And that's what collects the light. So the light comes in, it hits the little thylakoids in the big stack called a grana, and that's what's going to be used to capture the light energy, and then convert that energy into actual useful forms that the cells can use to make ATP for themselves, and eventually make sugar. So pretty cool way, to make sugar that will eventually be used to make ATP. So pretty cool way that plants do this, but chloroplast is essentially a photosynthetic bacteria that was uptaken and then just coexists now the same way that a mitochondria does. They're pretty cool. Endosymbiotic theory origin for those as well. Ribosomes, exact same thing that our cells do, that prokaryotes do. Our ribosomes are a bit bigger. They function pretty much the same way. They work the same way. They're found all throughout the cell, either bound to the rough endoplasmic reticulum or floating freely in the cytoplasm, and that's just kind of what they do. They make proteins. Every ribosome on the planet makes proteins. Eukaryotes have a cytoskeleton as well. Our cytoskeleton is a little more complicated. There's 3 things that make it up: microfilaments, microtubules. There's another one in there that's not here. Where are those? Intermediate filaments. Yeah, they're not on there here. But the cytoskeleton in our cells helps to provide rigidity to our cell. So a cell membrane, the same way that a water balloon is kind of squishy and blobby, our cells would have the same thing if they didn't have a little extra support structure inside of them. So this isn't totally solid, but it gives a little extra softness, so you don't just pop the little water balloon cell by looking at it incorrectly. So that's essentially what the cytoskeleton does for our cells. It also allows cells to move things around inside of them. So they have long interconnected filaments that run along the whole cell. You can use that as a highway. You can use it to move things around inside of the cell. You can attach things to it, move them around. You can use them to grab something and move it to. If you recall mitosis, this is essentially how that works. Really cool way of doing this. And the cells can also use the cytoskeleton and manipulate it to move around. So things like amoeba, that's how they work. They move their cytoskeletons around, and that's how they ooze from place to place. So pretty neat things that the cytoskeleton does. So check out this little chart. This is a comparison of prokaryotes versus eukaryotes versus viruses that tell you what they do and don't have in common. So pretty much the only thing that viruses have in common with cells is they have some sort of DNA or RNA inside of them, not both, and that's about it. So go ahead and check these out. You don't have to memorize these charts. So let's go ahead and chat real quick about the kind of a brief overview of eukaryotes in general, kind of some of the eukaryotes that will be commonly encountered in the world of microbiology. So the first one is fungi. Fungi account for quite a lot of infections every single year. Fungi are either going to be divided into 2 forms, either the fungi that you can see or the fungi that you can't see. So the fungi that you can see are going to be the ones that you think about when you think about mushrooms, fungus in general. So mushrooms in your front yard, the puffballs, the things that grow in the pasture that you go jump on and they send giant poofs of brown spores, smoke everywhere. Those are super cool. Gill fungi, you've probably seen those in your yard too. They look very similar to mushrooms. So things that you can see with the naked eye. More concerned about those if you eat them, kind of thing, or inhale their spores. But the ones that little of more concern for the microbiology world are the microscopic fungi, molds and yeast. So yeast live on and in our body. They're brewer's yeast, baking yeast, and things like that. Yeast infections. There's lots of different yeast on the human and in and on the human body. Animals and things like that. Molds. Molds live in, the ones that live underneath the sink. Mold is also a contaminant for microbiology. So molds and yeast are the ones that tend to be studied a lot more in the microbial world compared to the macroscopic fungi. The vast majority of fungi are unicellular. And that makes sense if you think about it. You can't see the vast majority of the fungal species that are out there. There's so many that we know about that are microscopic. The vast majority of them you can't see. The very few that you can see make up the kind of the minority of the overall fungus that we've identified. There's probably way more species of fungi that we don't know about yet that are all going to be microscopic just because you can't see them. So how are we going to find them? They're pretty neat. Fungi are super cool. So let's talk about that one real fast. So microscopic fungi. If it's a microscopic fungi, it's going to exist in either one of two different forms. It's either going to be a yeast form or a hyphae form. So a yeast form fungus is a nice little round ball. Looks exactly like a little round yeast cell. Yeast is just the generic name for round circles. That's not a thing, a species. Yeast is not a species. Yeast is just a shape. It's an asexually reproducing form. They do this. They bud and make another little copy of themselves off to the side. You can see that here. This budding little yeast cell, another cell is budding off of it. Eukaryotic asexual reproduction. And then they come in hyphae form. And hyphae form is essentially really long filamentous strands of cells. Instead of nice little round balls, you have really long, thin, skinny cells that string out, and they're all connected to one another. Either by cell walls, which we'll talk about in a second, or they're divided in different sections, which we'll talk about in a second. But you're going to find fungi in one of those two forms, either yeast or hyphae form, if they're microscopic fungi. And sometimes they can do both. Sometimes at a particular temperature, or if they're in the environment, they will be a yeast. And then if they're in a new environment or they get inside of a host where they want to be, they'll become a hyphae, and they'll change their form. So some species of fungi can do this. Not all of them can. And species that can do this are called dimorphic. They can revert from that hyphae form to that yeast form, and then back to the hyphae form, depending on temperature or environmental changes and things like that. So pretty neat. That's what allows some particular molds to become pathogenic, to cause disease in humans. They live perfectly fine in the environment, and then when they get inside of you, they can change their form and then do something else to cause disease. So really interesting how they do this. So fungus. Fungi are all heterotrophic. There's no photosynthetic fungus. They look kind of like plants, the mushrooms in your yard, but they're not. They cannot make their own food. They have to get food from some other source. The vast majority of fungus that are out there are harmless little organisms that eat decaying matter. They break it down like a spider. Their cells secrete digestive enzymes. Little digestive enzymes break down, the dead animal or the wood that they're sitting on or whatever it is, or in this case, your feet. They break down that skin, and then the digestive enzymes will be sucked up by this organism's cells, and then the nutrition can occur inside of the cells once they've pre-digested everything outside, and they suck everything inside. And that's how funguses eat. So sometimes when they get on us, they do the same thing. So you can see here this guy's got athlete's foot, this person does, and then the fungus that's growing on his toes are eating his feet. They're secreting digestive enzymes out into his skin that are breaking down the skin, making it rough and red and itchy and painful. And then they're sucking back up the digested skin particles, his cells, and eating it. So that's how they kill you, is essentially what's going on. They break your cells down. The vast majority of fungus out there don't do this as parasites. They're out there just breaking down, rotting wood or dead animals, things that they're supposed to do, rotting fruits. But sometimes they have the ability to get in or on us and then eat us down, break us down. So that's essentially what's going on here. So if a fungus causes an infection, it's called a mycosis, a fungal infection. The toxins that fungus can produce sometimes are called mycotoxins. So myco is the prefix for fungus. So a mycologist, someone who studies fungi. So fungi are pretty cool as long as they're not growing on your feet or somewhere else where they shouldn't be inside your body. So yeast are going to be nice little round, soft, squishy looking balls, and that's essentially what makes a yeast. That's the characteristic yeast shape. They look like little soft, little soft round marshmallows. Is essentially what they are. And as I mentioned, they reproduce by that budding concept. The little cell will have a little smaller cell pop off the side, and that cell will get bigger and bigger, and then another one will pop off that. I mean, you can see that here. All these little budding yeast cells getting ready to reproduce. So they're ready. It'll bud off. The nucleus will replicate the DNA. A little smaller piece of the DNA will go in there. That will copy the original cell, retains one. And then that's what's going to happen. They get smaller and smaller and smaller until eventually you get down to tiny yeast cells that really aren't going to be able to do very much. So yeast are neat, but this is the way that they reproduce. There's thousands of different species of yeast. This is, once again, just the generic name for the shape, not an actual species. So filamentous fungus, the hyphae form, their hyphae essentially grow all on top of one another. It's essentially kind of like hair, so they just kind of blob all over the top of one another. They grow all out in the environment and things like that, out over your skin. And if you've ever seen ringworm, the little round ring, that's the hyphae that's growing out from wherever you got the original cell or the spore introduced to your skin, probably through a cut or a cat scratch or something. So they grow outwards. So that's kind of that's the hyphae that's growing out. That's the ring. So as the hyphae expand, they get larger and larger. They take in nutrients from the things around them. And hyphae are going to be either open cells, as in everything can kind of ooze together. All the different cells of the hyphae are all connected. The ribosomes can, the nucleuses and all that stuff can just kind of run between cells. They just blob all together. And then you can have cells which are divided. The nucleuses are stuck in their cell. They can't get through to the cell next to them. And that's useful to identify different species of fungi. So some of them are septate. That's what the term septum just means, divider, the septum in your nose. Things that it's just a dividing line, a divider, a wall, if you like. So septate fungi, that's a classification for identification if you might have one that's causing you sick or you want to know what it is. Or non-septate fungi, ones that aren't divided. And once you know which one's which, or you can identify which one's which, that's a good way to help identify what the particular species of fungi that you're looking at is. So if you see fungi on your petri dishes, and they're fuzzy or velvety looking or kind of hairy, that's the hyphae that you're looking at. So that fungus is in a hyphae form, and that's what you're looking at. So mold on a petri dish, yeast on a petri dish, yeast form fungi look just like bacteria for the most part. So you have to really know what you're looking at to be able to tell them apart. Fungal reproduction is essentially going to vary from species of fungi to species of fungi, but it all pretty much involves spores. They will have little spores that come out. The spores will go land in the environment. The spores will germinate, will grow a small little hyphae, which will eventually produce more spores, which can grow out and start the process over. So that's how that works. So either asexual reproduction is yeast, or these reproductive hyphae spores, depending on what type of fungus you happen to be. So lots of different types of mold spores that are found out there. You can take a look at these if you want to. This is how they identify partly the species of mold that's causing infections or is found somewhere, or what the type of spores that it produces are. Different types of molds, and pretty much every species of mold produces unique and different types of spores. So if you know what the spores look like and what the size of them are and things like that, shapes, you can identify the particular species of mold that's there. So that's a really interesting talent for people that can do that. So very interesting, very useful thing to know is the different types of mold spores that are out there. So sexual reproduction also occurs in fungi. They can mate. They can unite their hyphae. Their hyphae can undergo the uniting of zygote kind of thing, and shake up their genetics if they want to. So once the hyphae have bonded, they're going to form this thing called a gametangia. Gametangia will produce spores that are sexual this time, and are diploid, instead of just asexual spores that they originally would be, just through traditional fungal mating. So you can see here our little piece of bread. You see the reproductive structures poking up, and the hyphae all down underneath, all throughout the piece of bread, and that's what makes the bread look fuzzy on top. And then the little things that are sticking up are the reproductive structures. So if you see a mushroom in your front yard, that's the reproductive structure of the fungus, and the hyphae all down through the ground that you can't see. So different types of species of fungus produce different types of spores in different types of ways. So here's the different classifications of fungi. They're pretty cool. They're identified and classified based on the type of spores that they produce and where on their fungal body that they produce those spores. Phylum Zygomycota. This is bread mold. This is the stuff you see that grows the black bread mold on every single piece of bread if you leave it out too long. They're often common parasites of arthropods, bugs, and things like that. They'll grow in between the little cricks and bins of the organism's exoskeletons, and then they'll grow over the surface of the organism and make them look all fuzzy, and then eventually they'll eat them from the inside out, so it's kind of horrifying. Ascomycota. This is probably the most important phylum of fungi for humans because it contains penicillin. And also, a lot of these fungal members are toxic or pathogenic to humans. So really, really dangerous members in there. It also has yeast, which is cool. So our baker's yeast, our brewer's yeast, and things like that are also going to come from there. Basidiomycota, this is going to be the mushrooms. And when you think of a mushroom, a stereotypical little white mushroom in your yard, the white and red toadstool, amanita, this is what you're thinking of. The Basidiomycota members are all stereotypical mushrooms. Chytridiomycota, these are the aquatic fungi. This is the only aquatic members of the fungi family. These guys are extremely dangerous for amphibians. They are parasites for amphibians. They grow on the skin of frogs and newts and salamanders and things like that in the water. And eventually they will kill that organism, and they can kill lots and lots of amphibians in a very short amount of time. Their spores are flagellated, which is cool, to allow them to swim through the water instead of just float through the air, because they do live in the water. And then you have things that are called imperfect fungi, and they don't produce sexual spores. They can only produce those asexual spores, and that's what classifies them. So there's the different kingdoms of fungi. They're pretty neat. They vary from they vary again, and they're classified based on the different types of spores and where those spores are produced inside of that fungus's body. You identify a fungus by looking at it underneath a microscope and what you can see with your naked eye. You're going to have to use specific media that's made to culture fungus. They need different nutrients. Also, a lot of bacteria will grow very happily, so you have to get rid of the bacteria. So specific media is needed to culture fungus if you suspect that your patient has a fungal infection or you want to grow them in a lab. And then you'll look at how that organism's body's put together. Are they filamentous hyphae? Are they filamentous, or are they yeast form? Are they septate or non-septate? Do they have this type of spore or that type of spore? What do they look like? What color are they? And that's how you're going to identify a fungus. I mean, you can send it away for DNA sampling like everything else if you want to. But if you know how to do it and you need a quick one, you can look at all of those different traits, and that helps you narrow down and maybe even identify the species of fungus that you have in question. So fungi do lots of different things for us in the environment. Sometimes some really good things, sometimes some not so great things. If you know anyone, or maybe yourself, is allergic to mold spores, the mold that might live in your house or in the walls or something moisture problem related, you inhale the spores that those organisms produce so they can reproduce. And then you inhale them, you get them in your lungs, they aggravate asthma, they can sometimes trigger allergy attacks and things like that in certain people, so that's never good. You can get pathogenic fungi inside of your body that grow in your respiratory tract, your athlete's foot. Obviously those are the very obvious ones. And then things like blights and things that you might not necessarily think about on plants, fungus are also very responsible for. So molds and things that hit crops and things, corn smuts and things like that, wheat rust. that destroy countless thousands and millions of dollars of food products across the world every year, just from some silly little fungus that happens to get inside of either the plant while it's growing or destroys them while they're being stored. And then they do a lot of good things for us as well. So I've mentioned this already, that they break down dead animals and decaying plants, but they're one of the very few things that does that. So bacteria do it, but fungi are about the only other thing that does. So without them to break down the dead and decaying animals and decaying plant matter out there, we would be in a world of dead animals very quickly. We use them in a lab. We use them in agriculture. We use them in food production. Sometimes we just eat them all the time. So fungus are very, very useful for economic growth, food products, and genetic research, because you can make yeast. Their yeast are very similar to human cells in certain ways. Their genes work very similarly to ours. You can make vaccines in certain things, antibiotics in certain yeast cells and things. That's where we get lots of our antibiotics come from fungi. So lots of different cool things that fungi do for us that aren't necessarily bad. So fungi are very important for us to keep around and study. So they might do way more things than we know about. So there's a bunch of different types of infections that humans can get from fungi. Take a glance at this table. You're probably familiar with a couple of these things, but take a glance at them, some of the different types of funguses out there, and a little bit about them. So on to protists. So protists, for the most part, are going to either be lumped into algae or protozoa. Algae, I really technically consider them plants, but they're not really in the same category as plants per se. They are very similar to plants. They have chloroplasts. They're able to produce photosynthesis, but they do so in a slightly different way. They're almost always unicellular, and they sometimes are found in colonial forms, but they're plants kind of light. So that's why they get classified as a protist, because they're not really plants and they're not really a bacteria. They're kind of a bacteria plus, so they're eukaryotes. So they get classified as protists, that eukaryotic that does a little bit less than a fancy eukaryote, but a little bit more than a prokaryote. And then you have protozoans, are unicellular eukaryotes that are really simple. They're pretty much all going to be the same. They all eat the same way. They look very similar in shape and size. The way that their cells are put together are very similar. And that's what classifies you as a protist. They're very simple little organisms. So they're kind of strange. And they all get classified on the different groups of protists based on how their cells are put together. So algae. Algae are photosynthetic little guys that are found pretty much all throughout the environment in the water. Algae are mostly going to be aquatic. They can be found living on the ground, but they need a moist environment at some point in time. They just desiccate out and dry when they're on the ground. An organism's called nostoc. They have a cell wall like every other plant does. They can either be macroscopic, if they're large forms into things of like seaweeds and things like that, or microscopic. For the most part, that's what the large majority of algae are. They're microscopic. They're beautiful little organisms. The study of algae is called phycology. There's millions and millions of different species of them out there. They're really, really pretty. They have chloroplast, that has chlorophyll A inside of it, a couple of other pigments, very simplistic form of chloroplast. Some of them have flagella and some of them don't. But that presence of the chloroplast that allows them to produce, their own food, is what classifies an organism as an algae and not just a Protista member. So algae are the most, when they're living freely in water and things like that, they kind of make up the base of the food chain. Things eat them. So they produce the energy for the rest of the food chain. So the sunlight comes in, it hits their chloroplast, they take that sunlight, they turn it into food, and then all of the other aquatic organisms that are in that environment will eat them, or eat something that ate them originally, or eat something that ate something that ate them. And that's how the energy goes into the food chain from these guys in the aquatic environments and things like that. This is commonly what's referred to as plankton, or zooplankton, phytoplankton, and things like that. Phytoplankton is the more appropriate name for these. Lots of, anywhere between 40 to 60% of the air that you breathe on a daily basis is produced by these guys. They recycle the CO2 for us via their photosynthesis, because if you think about it, there's a lot more water for them to live in than there is surface land for trees to live on. So there's a lot more of these guys proportionally than there are trees to produce photosynthesis for us. And then sometimes when these guys overgrow, their toxins, they produce toxins, some of them do, that can cause things like paralytic shellfish poisoning when organisms eat them. So the organisms grow in the water, they produce toxins. The organisms like shrimp and things like that will eat the algae for food, and then the shrimp get the toxin inside of their body. The shrimp eat a lot more algae, the toxins get concentrated inside of the shrimp, and then you eat the shrimp, and then instead of eating a little bit of toxins, from maybe 1 or 2 algae cells, you ate 500 algal cells worth of toxin, because that's what the shrimp ate, and then now you're sick. So paralytic shellfish poisoning and things like that are caused by algal blooms, red tides, and things like that. So another interesting side effect from the overproduction of algae. So algae are really cool. They do lots of different things for us, and they're classified based on the different types of pigments that are found inside their chloroplast and the structures of their cell walls. And that's how they classify algae up. There are Protista. Protista is super neat. There are about 65,000 different members of the species Protista. They're all pretty much going to be unicellular. Colonial forms are out there, but they're not the most common things out there. They really vary in shape. They all lack cell walls, which is a characteristic of Protists. Most of these guys are absolutely harmless. They just live out there somewhere where it's wet or moist. But some of these guys can cause problems in both humans and animals. Malaria, African sleeping sickness, and things like that. Parasites that are protist that cause problems in humans. Every single one of these guys are heterotrophic. They cannot make their own energy. They don't have chloroplast. They have things. Some of them have non-functional chloroplast, which is really weird. So interesting little things that these guys. So they don't have real chloroplast. They're I can't think of the word off the top of my head. But they're non-functional chloroplasts. So very strange things that these organisms have and that they do. So these guys just swim around their world and they eat stuff. And that's how they will function. They move around, they eat other microbes in their environment. They'll suck up an organic matter that they can find, and that's what these guys do. So there's different types of protozoa, and we'll talk about those in a second, but there's 2 different forms of protozoa that are out there. You'll have protozoa that are mobile and protozoa forms that are not, and that's essentially the different forms. You have a form that's able to move around and eat and swim around in the environment, and that's called a trophozoite. They're going to have either a flagella, they're either going to ooze from place to place, so a pseudopod, fake feet like an amoeba, and they're going to have those little cilia which they swim around from place to place. If they're moving, they're in the trophozoite form. That's a moving form that can eat and take in nutrients from the environment. And then what happens is occasionally they will enter into an environment that's not particularly happy for them. Either it's too dry, there's not enough nutrients, something that they don't like about that particular environment. And then that protist will enter the cyst stage. And a cyst is a very highly resistant, dormant form of a protist. The organism is essentially hibernating inside of there. It's going to take a lot to destroy these protocysts. They can survive a lot of water treatment plants and things like that. So they, essentially he'll stay like that forever until the environment becomes good again. The nutrients returns or whatever, or maybe it enters into a new host, and then they'll break open the cyst and enter back into the trophozoite form. So cyst formation is a very interesting way that a protist can transmit infection around via water. Different parts of the United States have different little fungal infections, or protozoal infections, that pop up in drinking water due to these cysts. So strange little thing. These guys almost always reproduce asexually. They all can. but some of them can also reproduce sexually. So it's not a huge thing to classify them based on how they can reproduce sexually or asexually or not. Here's the classifications of protists. It's based on how they move around, based on how they reproduce, either sexually, asexually, or both, and how their life cycle works around. So Mystigiopora, Sarcodina, Ciliophora, and Apicomplexa. So Mystigiopora, these are Mystigioporas. This is Giardia, Giardia lambia. So Mystigiopora, they are going to mostly move around with their flagella. There are some that move around like an amoeba, that kind of oozing thing. They're all going to reproduce by sexual reproduction. So Sarcodina, there are stereotypical amoeba. You can see them here that look like the little oozing guys that move around. They're going to asexually reproduce. They just split into a new organism. And the vast majority of them are going to be swimming around freely, chilling out in the environment, just doing their happy little thing. So Ciliophora, they have cilia, hence the name. You can see them here, floating around inside of their little, floating around outside of their cell, and that's how they're going to move around. You can see their little gullet here, their little oral groove, where they're going to move their food in. So Ciliophora, and our last one is Apicomplexa. So these guys can't move around except for their male reproductive form. So these guys are super weird. They're all parasitic, which is another big highlight for these, which makes these guys probably very primitive in the form of protists. They have very simplistic structures. Parasitic organisms tend to be fairly primitive as well, and they can't move. So really strange little guys here. But the vast majority of our problematic protists are going to be these guys. So here's our parasitic ones. You can see down here the really bad ones. All the super scary ones are down here, the apicomplexans, malaria, things like that. But there's still some really bad ones up in the other ones. Apicomplexans and things like that. Entamoeba, brain-eating into amoebas and things like that. Sexually transmitted diseases, trichomoniasis, things like that. Sleeping sicknesses, leishmaniasis, and things like that. So some really fun pathogenic protists that are out there that do really diverse things in humans and animals. So a couple of really important ones are trypanosomas. So trypanosoma is a protozoan that's found in South and Central America as well as Africa. And they're spread by insects. So the African version is spread by the tsetse fly. It's called African sleeping sickness. The organism is Trypanosoma Trypanosoma brucei, switch those. So Trypanosoma brucei is spread by the bite of an African tsetse fly. It takes a blood meal from someone that's infected or an organism that's infected with these little trypanosomas swimming around in the bloodstream. It flies to a new person or a new animal. It bites them, and the little protozoan swims down the tsetse fly's proboscis into the new blood, from the blood of the old organism into the new organism, and that's how the infection spread around. Chagas disease is spread in South and Central America, parts of Texas, by the assassin bug. Essentially works the same way, except these guys poop around your mouth, which is a little scarier. They can also bite as well. So there's a couple of different ways for this particular organism to get you. Sleeping sickness, you essentially fall into a coma and you don't wake up. Chagas disease makes your heart and internal organs and arteries and things essentially harden. Holes start to form in them and things like that, and you essentially just have a heart attack. So horrible ways to go for trypanosomas. Entamoeba histolytica is an amoeba that is spread around by contaminated water. Amoebic dysentery is another name for this, very commonly found across the world from contaminated water through fecal contamination by humans. So it cysts up in the water. It doesn't care if it's treated water or not for the most part. You drink the water, the cysts get inside of you, they open inside of you, they insist inside of you, and then that's essentially what happens. They set up shop in your intestines, and then you pass them through your feces, and you don't have a really good time while you've got them inside of you. So helminths. So our helminths are going to be our parasitic worms. I mean, if you recall, our eukaryotic worms are only going to be talking about their reproductive forms in the sense of their eggs or their larvae and things that are tiny that we can't see without a microscope. So the big guys get tossed in here as well because they're part of the cycle. But the little ones are why we lump them into the field of study of microbiology. So parasitic helminths, little flatworms and things that are swimming around out there, like planarians, we don't really much care about them. But the ones that get inside of people, those are the ones that we care about, and animals. Because they parasitize our tissues. They're either going to eat our cells outright, they're going to break our cells, or they're going to siphon nutrients away from our body through our digestive system and nutrients and things like that, which directly harms us as well. So not the best thing to have inside of you. So a lot of these things occur from eating, coming in contact with the cyst or the eggs in the environment through your diet. You get them on your hands or on the food that you eat, and you put them in your mouth, and that's how they enter into your body, or they're already in the food or on the food when you eat it, and that's how they're going to move around and get inside of your body. Essentially, all these things are is giant sex organs. So it's essentially a body that's designed to grab a hold of the intestines or some sort of tissue inside of you or another organism, and then the rest of it's all just reproductive organs to make tons and tons and tons of egg and sperm at the same time. So these guys can get inside of you and then instantaneously start to reproduce really, really, really quickly. Once their eggs are fertilized, either the eggs are going to be passed through the feces, you will come in contact with the eggs will hatch inside of you, start the process over again, or the little baby larvae will hatch inside of you, and then they will either travel out through your body and cyst up inside your tissues and things like that, and having people get tapeworms and things like that, or they'll pass through your feces and things like that, and then enter the environment and then find a new host that way. So real fun ways that these organisms do this. So you got two different types of parasitic helminths. You got flatworms and roundworms, and that's exactly what they sound like. If they're flat or if they're round, and that's how you break them apart. With our flatworms, you got tapeworms and trematodes, things that are flukes or are tapeworms. So our cestodes are, our tapeworms. Cestodes are super weird. They are going to be, both of our little guys here, our worms are going to be identified based on how they're put together, how they reproduce, their shape, and things like that. The different types of larvae, the worms that they reproduce with, or what the larvae look like, what the adult organisms look like, the eggs look like, what the shape, how many eggs they have, what the shape of the eggs are. All these different things matter. How many lobes of the ovaries there are, how many lobes of this there are, how many lobes of the testes and things like that there are of these organisms. And that's how you identify different forms of helminths and different species and things like that. It gets very difficult to identify these guys. So there are about 50 different species of parasitic helminths that are important to humans. They're found all over the world. The vast majority of them are going to be transmitted through food. You're going to come in contact with or water. You're either going to come in contact with the larvae or the eggs from the water or the food. So the food that you eat has dirt on it, or the organism is already inside the food, and you bring it into your mouth and you eat it, or you drink some water that's got them on there. Or they're going to get inside of your body from some sort of vector, like an insect, like a mosquito, or the tsetse fly, or a tick or something that's going to puncture your skin and then insert that parasite for you inside of your bloodstream. So once that organism is inside of you, they can set up shop, and then they can start to feed on you. So billions of people across the world are afflicted with parasitic worm infections, simply because of lack of hygiene or contaminated water and things like that. One of the most common ones that's found even in the United States are some things called pinworms. And pinworms are a very interesting little form of worms that are spread around, mostly through little children and small children and things like that, simply through contact, because little kids are kind of dirty. So this organism, you come in contact with it through the feces of an infected individual. So you get the feces on your hand. The eggs, essentially, is what happens. You get eggs of this organism on your hand. You put them in your mouth. The organism can then hatch inside of your body and set up shop. So how this works. So little babies, when you diagnose this, it's very interesting. So little babies scratch themselves all the time. They touch themselves all the time, and they also put their hands in their mouths all the time. So that's exactly what this organism preys upon. So once a pinworm infection sets up shop inside of a person's intestines, a little baby's intestines, at night the female eggs will come to the rectum, the anus of that sleeping child, while the baby's asleep, stick her little reproductive tail out, and deposit her eggs around the surface of that child's anus. Now, there might be 4 or 5 pinworms female that do this. And the eggs are really itchy and really irritating. So the little child will reach back there and scratch them when they wake up to try to get the itching to stop. And when they do that, they get the little eggs all over their hands. So then they stick their hands in your mouth, their friend's mouth, and things to spread them around, and that's how the cycle continues. So to diagnose this, you stick a little piece of tape over your baby's bottom, and in the morning you pull it off, and there will be pinworms stuck to it. And then you can go to the grocery store and get some Pin-Away and treat the little infection. Very common form of parasitic worms that are found even in the United States. In fact, there's some theory out there that some people out there, theories I should say, the light use of the word theories, that actually purposely infect themselves with pinworms to help fight allergies. And we'll talk about that a little later on when we get to what causes allergies. So this is a little brief overview of our eukaryotes, kind of how they're put together, how they function, what they do. So if you guys have any questions over this PowerPoint, or you want me to elaborate on anything or talk a little bit more about anything, or you just have any questions, feel free to let me know. If not, have a great rest of your day.